Okay, let's continue now with pictures. So here's our little picture. There's our cloverleaf, which you remember is kind of the same thing as the iRes. Yeah, same structure, just stuck to a different thing. Yeah. It'd be nice if they could just call it one name. I guess it's a cloverleaf with iRes function. Yeah, maybe that's how we rationalize it. So here's the same thing in pictures that we just talked about in words. The cloverleaf is going to be formed with the help of 3CD and poly-C binding protein number 2. The poly-A binding protein is going to bind the poly-A tail. So this is the genome, and so these two are going to help the cloverleaf form. This one's going to help the poly-A tail just be its best self, and it'll also protect the poly-A tail because nucleases would try to cut from the poly-A tail from the 3' end to the 5' end. And poly-A binding protein's main job is to prevent that from happening. It's a protector, a general purpose protector and server of RNAs. So there you go. Like a private security firm, I would say. All right, so now that we've folded this up, we're going to circularize. So here we are circularizing, and the poly-A binding protein is going to link both of these together, but it needs help from poly-C binding protein and 3CD. Cool. Now a second 3CD and maybe a 3D as well are going to actually synthesize the primer, and so the little green dot is a VPG, and we're going to add 2 U nucleotides to there. We're going to do that by that sort of sliding. We're going to put it on the first A, then slide it to the second A, and then add another U where the first A was. And if you want that in words, just go back a step. And if you want that in pictures, go back a couple of slides. It's there. Yeah, so that should help you. Now that we've made this little primer, VPG with 2 U's on it, that can go down to the poly-A tail at the end of the virus, and it can be extended. And the 3D polymerase is going to do the extending. So everywhere you see this sort of red little crescent, that's 3D. And everywhere you see a blue Pac-Man inside of a red crescent, that's 3C and 3D together. And remember, here is 3C, here's 3D, and together they are 3CD. Good job, guys. All right. Okay, that was making a minus strand. Yeah. Basically a whole bunch of setup, a whole bunch of weird stuff to make a primer, and then 3D just like extends the primer and copies what's in front of it. Yeah, 3D, very boring protein, boring and predictable, the way we like our proteins, right? Now it's got 2 strands that are together. Oh no, because it needs to pry those apart and it needs to copy them separately. It needs to make more minus strands and more plus strands, and it needs those 2 strands separate to do that. You know what? It's the same proteins once again. So you're going to have your poly-C binding protein number 2, poly-A binding protein. Those are going to come together. Those are going to separate out these strands because they're going to bind to one strand specifically. Human ribonucleic protein C and the 2C ATPase, which is a helicase-like protein. Those are going to bind to the other strand, and so these are binding the minus strand. These are binding the little plus-strand genome. And so now we're going to reposition. So remember, wherever you have these little orange and gray, that's going to be marking the plus strand. There are a couple of nice little nucleotides right down at the end of that. There are 2A nucleotides. And so those are the things that are going to get copied. We'll put this primer on there. It has 2 U's. The U's are going to bind to the A's, and then we're going to extend this and copy all the way to the end, and that's going to be the 3D polymerase that will do that. The neat thing about this is that because you've got 2C and RNPC on here, those will keep sort of peeling off the strand you just made from the strand that, from the template strand. And so you can get lots of these on there. And as many 3Ds as you have, you can get them all lined up. And so each one of these has a 3D, and each of them is copying along. And so they'll be all going in a row, generating like copy off the same template at the same time. It's just a way of copying multiple copies, and they call this a replicative intermediate, with multiple plus strands being made from one minus strand. And each of these 3D polymerases is able to just peel off the copy that's in front of it. So once that starts, it's easy enough, apparently, for the polymerase to do this. So it's like the polymerase is acting as if it was a helicase. It's not a helicase, for sure, but it's kind of doing the job of one. So, yeah, that's the idea. All right, other cool facts. The 3D polymerase will actually form these lattices. So each of these little blobs in here is one 3D polymerase, and they lock together, and people have shown that these things exist inside of cells. We don't know why, but since picornaviruses are going to be replicated near or on membranes, we think these are actually what is maybe on the inside of some of those membranes, and so this is going to be like a proper replication factory with lots and lots of little proteins. Now, to get this many copies, you'd have to translate that genome a lot of times. And one genome can be translated many times. And then once the genome is copied, you'll have more strands that can be translated, and you can make more and more. And so, yeah, eventually something like this becomes possible. The 2C protein, which is the helicase-like protein, has two interesting features. One, it works like a helicase, we think, in that it has helicase motifs, which, remember, helicases are not proteins that unwind DNA or RNA necessarily. They're proteins that really want to slide along one strand, and they'll push another strand out of the way to do it. Yeah, step over their own mother to slide along RNA. That's a helicase for you. Yeah, so they do that thing. They do unwind, but unwinding, think of that as like a secondary function. Mainly, they are single-strand sliding proteins that just sort of snowplow that other strand out of there. And so this one will actually interact with membranes, and in the center, that's where all the helicase domains are. And so what you've got here is, like the pore in the coronavirus double membrane vesicle, polioviruses make single membrane vesicles, and we think that 3C makes the pore in the single membrane vesicles. Yeah. So it looks as though it would be a hexamer based on the structure. And yeah, it's probably going to be involved in loading components into the replication factory and spitting completed copies back out of the factory, possibly with help from the ATPase. Domains of the helicase. There you go. Okay, and that's a good place to stop this section. We'll come back and pick up a couple extra pieces on assembly that you may not have had, and then we're good. We will be good. So good.